Okay, so now it's time to talk about those fabled neuroglia, the support cells that do so much to aid the neurons in their function of producing action potentials. So, turns out there's about 1 trillion neurons or so in the nervous system, but amazingly, neuroglia outnumber the neurons by somewhere around 50 to 1, maybe, but that may be a little over exaggerated, but nonetheless, by quite a bit. Okay, so we call them glia. Can just go shorthanded, just call them glia, and that'll be perfectly fine. Their main job is to support, protect neurons, and they're going to help provide this overall framework and mass that we call the brain and spinal cord. Okay, so neuroglia count for about half of the brain's volume. It's pretty impressive. You think about the brain weighing about 3 pounds, and half of that, weighing about 3 pounds. So most of that's going to be neural tissue, but the volume of the brain, over half of that is neuroglia. There are 4 types of neuroglia found only in the brain and spinal cord. The first type are the astrocytes, and we'll talk about each of these in more detail here in a couple slides. Ependymal cells, microglia, and oligodendrocytes. Now here's a somewhat overwhelming picture of these cells, and we can see here the cell called an oligodendrocyte. It's going to have largely a protective function associated with it. Astrocytes that seemingly are part of the overall framework support. Microglia that are also part of the overall, over here, that are part of the immune system, as we'll see in the brain. And then these ependymal cells that are so critical to the function of our brain and spinal cord. So let's look at each of these individually. Astrocytes are pretty much star-shaped. If we go back and look, you can see this star-shaped appearance. And what happens is their cytoplasmic processes will extend and provide anchors on both the neuron and on surrounding blood vessels. And the feet that surround the blood vessels can eventually form an entire surface that covers the whole blood vessel. And then this results in what's called the blood-brain barrier, because you remember from when we discussed the 3 types of junctions in between cells, that tight junctions are those like a needle and thread, and they sew up the membranes together so that no fluid can pass between those cells, and therefore anything that wants to enter the brain has to go through the epithelial cells that line the blood vessel, or through these particular extensions of the astrocytes. So this is all part of the blood-brain barrier. Ependymal cells are cells that line a structure in the brain called the choroid plexus. And the choroid plexus is found in areas of the brain called ventricles, and they secrete a substance called cerebrospinal fluid, or CSF. And this is what the brain and spinal cord sit in, bathe in. It carries waste, it carries nutrients, it does all kinds of great stuff. So CSF is absolutely vital to the operation of the central nervous system. In addition, these ependymal cells can serve as stem cells for neuron development. Pretty exciting field that isn't very old, 10 to 15 years ago, that they certainly discovered that stem cells existed in the brain and would and can develop into neurons. Microglia are just types of white blood cells called macrophages, specialized types, and they're basically trapped in the brain during embryonic development. They're white blood cells that get trapped and stay within the brain for the rest of your life. So they're going to help out with inflammation. They're going to help the CNS get rid of stuff that shouldn't be there. But nonetheless, you should understand that infections in the brain are very, very serious. Encephalitis, meningitis, those conditions are very serious just because the brain and spinal cord don't appear to exhibit a very robust type of immune system. Oligodendrocytes are really spectacular cells in the central nervous system, and they create extensions, quite extensive extensions, that will surround axons and protect them and bundle them into nerves. These wrappings that they can perform around, that can form around axons is called myelin. And so one oligodendrocyte can form myelin around many neurons. And we see these cells beautifully illustrated in this picture. The astrocytes over here that are extending their feet-like processes, and so many processes will cover that there won't be any evidence of the external portion of that blood vessel available. Microglia we see, and the big old oligodendrocytes. And then lining here, there are the ependymal cells. Now, there are two types of neuroglia found in the peripheral nervous system. The first type are your general vacuum-made type cells that are going to just take care of the general cleanup of the area of any neurons in the peripheral nervous system. So they're going to help regulate that environment. The second major type of cell is called a Schwann cell, and its overall function is extremely similar to the oligodendrocytes. The oligodendrocytes, remember we just covered, are to bundle axons, protect them, and form a myelin sheath. And they can do this around several different axons. Schwann cells can't. They can only form a myelin covering over one axon at a time. And again, this can be called the myelin sheath. While they can't cover multiple axons, they can still organize multiple axons, but it won't have what's termed the myelin sheath around them. We'll see this in just a second. Now, these cells can line an entire axon, and as they do so, they will form these long areas of this myelin sheath. But in between each cells, there are these open bare spots called nodes of Ranvier. Okay, we can see the PNS axon. So this is a Schwann cell beginning to surround and form myelin sheath around a single axon of a neuron. All right, so when we see myelin, it appears glistening and shiny, and that is termed white matter. So white matter is associated with myelinated axons versus gray matter, which are the cell bodies, which aren't ever covered in myelin, and any axons that aren't myelinated. So those would make up what's called gray matter because they appear dark and dingy under a microscope. Now, also you can see this notion of organizing and bundling several axons. So that can happen with the Schwann cells. These aren't myelinated then, but they are organized into a coherent arrangement. And you can see the myelin sheath that surrounded this axon, beautiful covering. And then up here we see the organizational aspects of Schwann cells as well. So here's a Schwann cell that forms a myelin sheath. Here is a Schwann cell that looks just like the illustration to the left. So a little bit more about myelin. It's insulating. Remember, we're conducting electricity along this nerve fiber, so this will help insulate it. And remember, the cell types are oligodendrocytes in the central nervous system, Schwann cells in the peripheral. And the myelin is actually the plasma membrane of these cells, of these oligodendrocytes and Schwann cells. And it turns out it's only a little bit of protein, but tons of fat, right? Because we know that a plasma membrane is mostly phospholipids, right? So that's going to make up the majority of the myelination. Okay, so myelination is the process of forming the sheath. Starts very early in embryonic development, and your nervous system grows very rapidly during embryonic development and doesn't complete actually till late in your adolescent term, right? So you can see how in your development, fat is a necessity for formation of proper myelination. So here we see the myelin cells. This is a Schwann cell. And then you can see this bare spot in here. This is the node of Ranvier. I wish I had labeled it, but I didn't. Okay, so CNS, it's oligodendrocytes that produce the myelin sheath. In PNS, it's Schwann cells. So myelination, what does it do? Why is it important? Well, it's going to increase the speed. If I stub my big toe or I cut off my big toe, something really horrible, then I'm going to know about it very rapidly because most of the neurons, axons, are going to be myelinated, and the action potential will actually move much faster through a myelinated axon than it does through an unmyelinated axon. Okay? So this makes the nerves appear as white matter. We'll talk more about nodes and internodes when we get into how electrical signals are conducted. And we'll get into all these terms in much more gory detail. Beautiful set of shots regarding Schwann cells and eventual myelination. Alrighty, so I just wanted to show you an organization, how highly organized it is in the brain between gray matter and white matter. And here's a slice through a spinal cord. And all out in here is the white matter. And then you see the you have to use your imagination sometimes, butterfly-shaped gray matter. In the brain, the gray matter is concentrated in this outer shell region, a lot of white matter in the midst of the brain, and just a few gray matter regions found right in this area of the brain. So our summary for neuroglia, we have our four neuroglial cell types right here. Nice, great summary. I invite you again to look at the face-to-face PowerPoint that I provide you to get more details and to observe this on a longer time scale. It's great. We'll see you for the next video.